Good afternoon, everybody. Thanks to you, Cody and Rosemary, for inviting Occam's to present here. I'm Paul Halpern, the co-founder and CEO of Occam's Bio, and I'm very excited to be here. My contact information is in the chat. So Occam's Bio is about generating best-in-class fast follower antibodies. Fast follower drugs historically outperform originator molecules in patient outcomes and profitability. But this historically validated paradigm which is standard practice for small molecules, has been inaccessible in the antibody space. And we're changing that using our proprietary antibody screening methods that allow us to build and test millions of variants around a predicate antibody molecule. So what we're doing in the first application of this method is to start with a leading, recently approved blockbuster antibody therapeutic. Use our system to build and test millions of variants and generate an improved proprietary molecule that we can patent and take through to the clinic through phase 1, 2, and 3, etc. These best-in-class fast follower therapeutics will improve treatment, reduce cost, and enhance access to life-saving medications worldwide. And this is who we are. The method I've just mentioned is invention of Dr. Scott Desane, our founder and chief scientific officer. His last startup was predicated on his last major antibody discovery invention, and that's Immunome, which is now a public company with approximately billion dollar market cap. He is a researcher at the Lankenau Institute for Medical Research, where he created this technology, and his background includes Yale, Dana-Farber, the Whitehead Institute, and etc. My background is that I've been an investor and business improvement expert for over 20 years, and I joined Scott to spin the Occam's technology out of Limmer into this business in 2019. We're also joined today by Thomas Halpern, a decorated Marine who is in charge of our strategic planning and project management. So here's what we're about. Here's the opportunity for Occam's. The market for best-in-class fast follower antibodies. Everybody's aware that the antibody business is growing to exceed $500 billion in worldwide revenue within the next decade. And the molecules that we are targeting, the very first one we have working in the lab, has a market value of over $10 billion. The 9 molecules that we are planning to finish in the coming 2 years before the end of 2025 have a combined value of over $50 billion. So that's the opportunity that we are chasing. How are we able to do this? This is the best-in-class fast follower strategy for antibodies. The critical thing is that last year, the Supreme Court verified for everybody that improved antibodies not cited by sequence in a patent are novel IP, and their discoverer can get their own patent. These decisions open up the antibody field to improvement, but only with a method capable of searching the literally tens of millions of permutations surrounding any predicate molecule. Legacy methods can't do that. But the Occam's method, our ultra-high throughput technology, can and has, opening up the field of best-in-class fast followers in the antibody space. So our method allows us to target these high-value antibodies, the most valuable, newest therapeutics on the market. These are drugs with long remaining protection from competition and a simple mechanism of action that are readily amenable to material improvement. And the public domain knowledge from the approval of the predicate molecule significantly de-risks the approval process for our antibodies. And the existing market data, which I've described some of, ensures the economic potential for our molecules once we succeed. So we're specifically planning to develop for out-licensing or collaborative development 9 of these best-in-class fast followers by the end of next year in a range of therapeutic areas, including oncology, human metabolism, autoimmune, and infectious disease. And here's where we are with our first 4 of the 9 projects. We started this path in January of this year. and in less than 6 months on our first molecule. The predicate in that case is an antiviral that generated over $400 million of revenue in the first 3 months it was on the market. We have been through our screening process, our library screening process for the first library, and the construction and screening of the second library and in vitro validation is ongoing. I'm pleased to announce that literally today I learned that the lab has validated that 12 of the sequences from our first generation library outperformed the predicate in a binding assay. Our second molecule, OB2402, is in the human metabolism space. The predicate there was the subject of a $2 billion transaction while it was still in phase 2. We are presently doing analysis of the first generation library created off of that predicate. Our third target is in the oncology space. It's a drug that is working its way up to first line in blood cancers, and that had 2 years ago revenue of $400 million a year. Our fourth target is going to be in the autoimmune space, and we're still doing the literature review on that. So the libraries, our first molecule will be validated in August of this year, the second in September of this year. So at this point, people say, how do you do that? So we're going to turn a little bit to the science. Basically, this slide compares our method to other antibody screening technologies, showing why we are uniquely able to access this fast follower antibody opportunity. In a nutshell, only Occam's checks all the boxes. Our fully mammalian system assesses antibodies and antigens in their natural configuration in a high throughput, cost-effective system that is comprehensive, which means it gives us data about every single antibody in the library that we test. And here's a little cartoon of how it works. Here we have an ordinary 293 T cell in a polyclonal population secreting its antibody. And each cell in this Petri dish will be secreting a different antibody. And every cell will have this anchor. We introduce a linker, and that allows us to create a complex of all and only antibodies being secreted by this cell on the surface of this cell. With that capability, we can introduce fluorescent antigen to literally label the cells that are positive for the antigen of interest with the antibody that they are producing. And if you look at the micrograph at the center of this slide, you can literally see it working, where positive cells are showing the fluorescent signal and cells physically adjacent to them are showing up as negative. So this micrograph is a really good visualization of both the sensitivity and the specificity of the Occam's method. And so we're using this to screen large, that's large 10,000 to 10 million library size. We take these synthetic DNAs, we put them into plasmids, transfect them into cells, and run them through our screening process 3 times, doing next generation sequencing on the populations at every stage, and using the enrichment from the screening as our output metric. So that is, in a nutshell, our discovery cycle. And what you can see here on the right-hand side of this slide is our demonstrated commercial success with this effort. We started with, I believe it's this one, a therapeutic quality antiviral antibody. We ran this through our system. We generated a cocktail of improved molecules. Of most important, we produced a molecule that had an improvement in neutralizing titer of over 2 times, which is therapeutically meaningful, to say the least. This slide shows the project plan for developing, on the top, for developing a single molecule, from selecting the molecule here at the beginning of month 1 to naming and isolating the lead candidate at the end of month 6, followed by 2.5 months of validation science. So this 9-month cycle And you can see from these numbers where the first 4 molecules I discussed earlier land in this cycle. Is being performed by our lab today with 3 full-time scientists and our inventor, Chief Science Officer Scott Desane. But that doesn't consume the entire lab bandwidth. In fact, we can run 3.5 of these projects at the same time. And that is, in fact, exactly what we are doing at this moment. We are running 3 of our projects and 1 project for a commercial customer as we speak. So here's where we stand. We have raised over $2 million to date, about half of that in equity and debt, with the other half in non-dilutive sources, an SBIR grant and commercial revenue. That has allowed us to put the infrastructure, equipment, and core scientific team in place to accomplish the plan that I've outlined here. And we are now raising $5 million. The point of this money is to provide for the completion of this program and operation of the lab long enough, 2.5 years, in fact, to allow us to start generating revenue, early revenue, from the out-licensing and collaboration deals of the 9 best-in-class fast follower molecules we're developing. About 60% of the money will go towards maintaining basic operations, payroll, rent, and licensing costs for the first 2.5 years of operations. And the other 40% goes to program-specific costs, such as reagents, outsourced lab work, consultants, and lawyers associated with each individual project. Here is a link, if you write it down really fast, to our patent and the original technology reference. And I would just like to summarize what we're about by going back here. We're deploying our proprietary technology in the development of 9 best-in-class fast follower antibodies in 2 years. These are improved versions of blockbuster therapeutics that will be capable of sustaining their own patents and regulatory approvals. And this is only the beginning. This Occam's method will continue to produce valuable, validated, optimized therapeutic antibody leads at this pace for years to come. So that's why we're so excited about it. I would love to follow up. As I said, I can take questions over voice or in the chat now, and offline, my contact information is in the chat. Thanks again, everybody, for your attention, and Cody and Rosemary for the invitation.